All right. So yeah, so this is going to be the phenotype. This is one that a lot of people are getting this semester, which I'm very happy for, believe me. So yeah, I don't really know what else to say about it. Remember, the genotype is talking about your genes for it. The phenotype is talking about what is expressed. All right, the 3 parts to a DNA nucleotide are, be sure you read all of the choice. Okay, so we know it's going to be deoxy, right? And the sugar is always going to be the ribose or deoxyribose. So hey, then you had to just know if it was basically phosphate or sulfate. And we've been talking about some phosphates lately, so hopefully you knew it by that. But yeah. Right, the DNA anti-parallel strands of nucleotides. So we're talking about a strand, each strand. How are those strands linked together? All right, so it's going to be hydrogen bonds. Exactly, they're holding the strands together. Of course, our nucleotides are held together in that strand by covalent bonds. Specifically, they're called phosphodiester bonds. Here's that by True or false? DNA polymerase builds DNA in the 5' to 3' direction. Okay, so this is going to be true. Most of us are listening to me keep saying 5' to 3', 5' to 3', and I hope that that's getting drilled into your skull because that's exactly the way you should remember it. So you heard 5' first, you know that's going to be basically BBK. So which enzyme connects Okazaki fragments that are formed during DNA replication? I don't know what this is a picture of. Sometimes I just pick the first picture that it suggests, and I'm just like, whatever, this looks cool. It looked like a badger. Yeah, it did. Okay, so the enzyme that's going to actually connect those Okazaki fragments together, it's called DNA ligase. And I know we only mentioned it very briefly, but it's actually going to be really important, not just in DNA replication, because obviously if you're making one strand continuously, which is our leading strand, and the other one's made in little segments, those segments being called Okazaki fragments, it's overall going to be the lagging strand. Those fragments aren't just going to spontaneously make bonds together. It always takes energy and effort to make bonds. So the enzyme that's going to foster creating those covalent bonds to link them back together is going to be DNA ligase. And it's pretty important for the stuff we're going to talk about today. Pretty good tool. So during transcription, what's going to happen? Now, this picture does apply to what's happening. This is actually a picture of transcription. I'll give you that. In transcription, RNA is going to be produced. And I'll write it up on the board here just so we can remember it again, because I know we're still learning this, and it's not that big of a deal that you guys aren't really like 100% on it. So DNA going to DNA, that's called replication. DNA going to RNA, called transcription. Remember, script is like writing basically the same language. So these guys which is nucleic acid, or let's just say nucleotides, right? That's their building blocks. And one of them is deoxy, and one of them is ribo. But that's just talking about looking at the original print in your Smithsonian Institution book versus your handwriting, like you're writing on it. Okay, that's that one. And then remember RNA to protein. Now proteins are written in the language of amino acids. That's their language. And this is called translation, from turning into a different language. We could even take it as far back as to say this is in our actual, for eukaryotes, anyways, it's going to be in our nucleus. For bacteria, it's just hanging out. If it's not in a nucleus, where is it? What do we call that? The cytoplasm. If it's not in a nucleus or in a membrane-bound organelle, it's in the cytoplasm. Okay. So bacteria don't have membrane-bound organelles, so it's always going to be in the cytoplasm. So if I ever ask you if something is blah 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 in bacteria, it's either going to be in the cytoplasm or on their membrane, which we will have explicitly talked about. Right. So when we get into metabolism, everything that happens in our mitochondria happens on their membrane. But that's why it happens in our mitochondria on the mitochondria membrane, because it used to be bacteria. So makes sense when you think about it. But anyways, it's your DNA. Here's your actual chromosomes, everything that's going to contain all of your genes. Now we want to take out the one working copy of your gene and turn it into protein. But you're not just going to have the one thing to direct all of that, right? You could have ribosomal RNA, which is rRNA. That's what the ribosomes are made up of. These are just machines that perform translation. Think of it as like if you guys plug it into Google Translate. If you're talking to somebody who don't speak that language, Google Translate itself would be the ribosomes. Or like that. All right, number 2, we have the tRNA. tRNA, or transfer RNA, what it does is it carries the correct amino acid, right? So I'm going to put, and this is tiny because it's loose in the back, and I apologize. Carries the correct amino acid, and it does that based on its anticodon. Has to match the codon of the mRNA. So that brings up to our third guy, our mRNA. This is our working copy of the gene. What's the gene? Our working gene. And this guy is made up of 3 nucleotide pieces. The same nucleotide, but now we can segment it into 3 as we're looking at it. 3, we put NT sequence equals the codon. Okay. Now down here, just to summarize, so you know what's happening with all of this during translation. Okay. The ribosome is going to bind first. Okay, that's 2. All right. Then what we can do is bring in tRNAs that have anticodons that are complementary to the codon on the mRNA. I'm going to put that. I just think it's not a non-wordy way of putting this up here. We'll say mRNA codon binds complementary C RNA and codon. For complementary, that means A to U or G to C. And that's the other way. Okay, that's what we're talking about there. So that's like the opposite of it. So if this one said A, then the other side of it would say U. That's what we're talking about. Or RNA. Then basically the last step of that, especially for our purposes for understanding what's happening before we can imagine the whole ribosome shifting and doing all its BS, we have to understand that the amino acid that is carried by your RNA binds to the amino acid next to it. So I'll say the tRNA amino acid 1 plus tRNA amino acid 2 creating a bond. Now that's called a peptide bond. And then it just keeps going on with that the whole way down the mRNA. Okay? So I'm trying to like hopefully note a lot still up here. We're kind of summarizing like pretty much more than half of the chapter 6. So I hope that that's helping some. We still got some questions on the review. So maybe that'll help. Right? All right. A codon is 3 bases of what molecule? Where's that codon itself? I'm going to circle those in different colors so you can just have visual separation. That stuff will stress me out. All right. So a codon is a 3 nucleotides on the mRNA. Remember how you can have the 3 nucleotides? It's still the same code, right? That you have as in the DNA. But in the DNA, we call it a triplet. We don't call it a codon. It's only a codon in mRNA. And that codon, that AAA or whatever, codes for a amino acid by association with the tRNA that carries it. Okay. All right. In what operons repressor proteins bind to operators unless a molecule that changes the repressor shape is present? This is some words, man. Okay. Everybody does terrible with operons. So don't take it personally. This is basically an operon here, all right? And an operon, all it is, it's a set of some proteins that you need to do a thing. It's usually more than one protein, and it's regulated based on the need. So in the front, we have the promoter. This part here. This is where RNA polymerase binds. So RNA, I'll just write it out so you guys don't ask a question about it. Okay, this is where RNA polymerase binds onto the DNA. Okay, so that's where it's going to start making its mRNA from the gene. That's what's happening here, right? That's how all mRNA is made. All mRNA starts with RNA polymerase binding at a promoter. All of them do. So in this case, our promoter is next to this weird junk. This weird junk is called an operator. And whether or not our repressor protein binds there determines whether or not RNA polymerase can go. So imagine it like this. This operator, otherwise don't care that it exists at all. That's just a region of the DNA, okay, the operator is. When your repressor protein, say it looks like this, okay, comes along and binds on the operator, it makes it so the RNA polymerase can't go. It's like a block, okay? So we have talked about two different kinds. We're gonna write this up here so you guys can know what that is. And that's the terms for these, no matter what kind of operon we're talking about, right? No matter if it is inducible or repressible, they have these always, okay? It is just based on whether it starts out on and can be turned off, or whether it starts off. And can be turned on, okay? So if you're on, you will get turned off, obviously when the repressor binds, right? So we'll just simplify this. This is when the repressor is activated. Okay. If you are turned off, you start out like this. If you're on, the black thing isn't there. If you're off, it starts out that way. Okay. So this case, we're going to turn these guys on. When that repressor protein is, are we activating it? No. In this case, we're deactivating it, right? To get off. So that you guys can think about this as the same exact types of steps going on, just basing it off though of whether we start out with the crap on it or not. Okay? So think of the crap is on it, the repressor is on it, that is off always. That's what that means. If the repressor is not on it, that is on. So if you start out on, this cannot have been there. So now you're going to introduce this. We'll say we're activating it to go stop, right? To go stand in the middle of the road to stop people driving or whatever. If you're already off, this guy's already standing in the road, waiting for the signal to go home. So in that case, once he gets that signal, now he's off work, he'll be deactivated, and now RNA polymerase can take over the shift, start doing its job. Okay, so it's the same kind of concept. It's just whether or not you're getting starting on or off. If you are on, you can be turned off, obviously, so you can be repressed. So these are called repressible. Okay. If you are off already, that means we're going to turn you on. If you're off and you need to be turned on, just like when you aren't in labor, but you should be in labor, we're going to induce you. So this is inducible. So that is the basic breakdown of what's going on. Now, in a repressible operon, usually the repressor will be activated with whatever you're making downstream here, like your product of whatever it is, the cell doesn't need it anymore. So it starts building up and building up. And that buildup is sort of like a signal to a factory line down the road. Hey, we're not selling any of these GAP genes. Stop making them. We don't need any more made right now. So that's the call that it'll get. And the guy says, yep, you got it, and pops the repressor on there. That call is usually signaled by the product, the genes in that case. So this is the arginine example that we had. You didn't need any more of that molecule because you had too much of it. So stop making it. That's what it said. The bottom one, the inducible, this was our last operon. We don't usually need to make the black glitter genes, right? We don't usually need to, but we've got a group coming into town that we know would love these black glitter genes. So we're going to go ahead and make the call to go ahead and start making those. So start making them for as long as we need to. All right? That's what these are. Don't overthink them. Just remember the terms. That's the hardest part. Hope that helps. Yeah. There's no, it's hard for everybody, so don't feel bad if you're having a hard time with it. Missense mutations. So we talked about the different kinds of mutations. How would you define a missense? Yeah, so much you guys got this one. It's going to be one amino acid for another. These are just terms that you're going to have to be familiar with, right? So this missense is going to be changing the amino acid. The nonsense is turning your amino acid into a stop instead. Whoops, right? And then the silent mutation is where you have the same amino acid, but the nucleotide in the background got changed, but it still codes for the same amino acid. So the protein doesn't really matter that much. At that point, it's not really affected, but you might check mutations like that anyways if you're looking at the actual genetics of it. Okay. And there's back mutations, which kind of speak for themselves. It's mostly for bacteria, though. Just going back to how it should have been. All right, it's a lot. Y'all are hanging in there. I appreciate it. I feel like chapters 9 and 25. 25 is a load of crap, if you ask me. We're going to talk about wastewater management, and the only reason we're even talking about that is because I did some stuff related to it in my old job, so you're getting dragged through it because of that. Otherwise, I don't really care about that chapter. Chapter 9 now, we're going to talk about your loving favorite, I'm sure, tonicity. So isotonic, hypertonic, hypotonic, movement of water across membranes. So that's probably the most important thing we are going to talk about that chapter. Just to let you guys know, to prepare you. So it's not horrible, but then we've got the metabolism chapters, chapter 10 next, and that one is a lot, but also is my favorite one, literally, actually. So, okay. Whew. Now that we definitely understand DNA, let's get into recombinant and like genetic engineering and stuff. So recombinant technology basically is referring to we basically learned from bacteria and the tools that they use to control what's going on in their cells. Stole those as well as some from viruses and all this stuff we learned about looking at genetics and how the cells manage that stuff, and hijacking it for our benefit. And that's really what recombinant DNA technology is. So some of the stuff that we can use it for, using DNA to identify an individual. Yes, I'm talking about CSI. Fixing the underlying genetic mutations that you may have or somebody else may have that could be potentially life-threatening to treat their disease in the active living adult person that has it. How about that? Permanently. How about that? Utilizing CRISPR technology to repair genetic mutations in a heritable way, which would allow us to change your mutations to help you with your genetic disease and keep it from spreading to your offspring. It's not real right now. Don't let anybody tell you otherwise. They can do this with like mice and rats, but it's like they cannot make it work for people yet. So I don't know. It might happen in our lifetime, but it is not a feasible thing yet. So don't hang it on your coat hanger as the miracle, upcoming miracle of medicine. Hopefully, eventually, but not now. So one of the things we have to understand about with genetic engineering is going to be DNA, which I hope that we've gone over at this point enough, right? So you know that your DNA, your two strands, your anti-parallel strands, that they are going to be connected to one another by those hydrogen bonds, like we mentioned. It's not very strong. So you can easily break those hydrogen bonds because they're weak. And we can do that with heat and cause your whole DNA genome to separate from itself using enough heat. And then if we cool it down, it'll just go right back to where it was. It won't mess up because there's so much relying on those A, T, C, Gs in whatever order and where they are, it'll just always go back to how it was, which is crazy to think about. But that's how it works, because it's reliable. So that's a useful tool for us. Another useful tool for us is restriction endonucleases. We also call them restriction enzymes. That's ACEs, right? Obviously, that's going to be an enzyme. Endonucleases, we're going to be cutting in the actual DNA code. Nuclease, cutting the nucleotides. Restriction, I say it's restriction. I don't know what they originally meant, but I say it's restriction because it's restricted to certain sequences in the DNA. These were enzymes that bacteria originally had to defend against foreign DNA to help them get rid of it. And now we have, collected them from bacteria and analyzed them and found out that they will each bind at a specific sequence and cut at a specific sequence. And all of those sequences are palindromes. What's palindrome? Same forwards as backwards, like race car. If you spell that out, it's the same forwards and backwards. Well, these are like that. It's slightly different, but you'll see what I mean in a little while. Okay. I feel like I should go over that first. Where's my pen? Did I not get it out? Yes. You're okay.
How do we see any of this?
I mean,
Like, how do we see it?
like the DNA?
Yeah, like in a microscope, we could just look at this.
Nobody ever asks that question, and I'm glad that you did. So if we're going to, especially if we're looking at the nucleotides, right? And I'm telling you like, oh, these are the sequences. How the heck did anybody ever figure it out though? So most of the time, this is going to be done by actually working nucleotide by nucleotide originally. So literally taking the chemical of whatever that nucleotide is. So adenine, guanine, and whatever as a nucleotide, and putting that in the mixture and then trying to build DNA that way. So with your DNA polymerase, because that is what has to add it. And how they, discovered that to begin with, I don't know how to answer that, how they got that far. But now that we have that, that is something that we can build off of knowing how that works, thanks to however the heck.
Right.
And I know the original pictures of the DNA double helix, that was done with like X-ray technology.
Okay.
Yeah. At the like atomic level, obviously. Right. And they say it's Watson and Crick. It was a woman, and they took credit for it.
Of course.
Yep. Now y'all learned a thing. Anyways, who is surprised though, especially back then? I should tell you guys at some point about Henrietta Lacks, if you haven't heard that story. Yeah, if you haven't heard it, it's worth knowing. But I don't really have the time today because it's a lot. But hopefully we have another time, remind me, and I will tell you. So anyways, this is going to be referring to the palindromic sequences. That's how I'm going to write this up here so you can know what's about to happen to you. All right. Your DNA being something like this, and it's literally just going to go T, G, G, C, C, A. Okay, and on the other strand, okay, that G to C, G to C, C to G, C to G, A to T. This is your DNA, right? And of course, we're smart, and so we know that this is your 5' and this is your 3', right? And they're going anti-parallel. Okay. Take another look at it. This is my palindromic sequence. Okay, so we'll start off with just the top strand. T, G, G, C, C, A. That's just the top strand. What does G bind with? What does G bind with? And what does T bind with? Interesting. Look at the bottom strand and look at it from the 5' to 3' direction. Now compare it to the other one in the 5' to 3' direction. They're the same. They both are like this. This is what I'm talking about with palindromic sequence. Crazy, right? It's almost one of those things that you're like, oh man, that's kind of crazy. Actual enzymes in nature that only pick up on these kinds of sequences. That's restriction endonucleases, made by bacteria to target these types of things, okay? Because they tend to be associated with things that they don't want in their cells. So what these enzymes do is they will cut between here, let's say, and because it's just on the other strand, it reads the same thing right here. So this is going to separate these. So now, if I were to get rid of this mess that I wrote here, where's my, there it is. Come on, guys, faster. All right, G, C, C, A, T. And then this would be like continue on with the rest of your double-stranded BS that's connected to it, right? And then the other one would be G, G, C, C, A. And then of course T is on the other side. And then it's connected to the rest of the other side of that. You starting to see what I'm talking about? But those can come apart because of those cuts. That's restriction endonucleases. So now we've got these weird single strand guys. They're still in a strand, but they're just single stranded guys that are sitting out here. We call these because, like I said, they're still hanging out with the rest of everybody else. We call these guys sticky ends because they want to bind back up with something. Could they just bind back up with that other brother that they just left just a second ago? I mean, of course they could. But if they happened to not to because of other things going on in a cell, then this would be detrimental. Because how could an organism or DNA that's supposed to have a function deal with that when it's been cut up weird like that, right? That's the thinking, I guess, of the bacteria. But the thinking of the molecular biologist is this enzyme, I don't know, EcoR1, this enzyme cuts this at this sequence. So if I put this sequence in a plasmid that I know what genes are in that plasmid because I designed it or bought it from Biorat or something, and I can cut it with EcoR1 and then have a gene that I have altered slightly from the human pancreas that is a gene for insulin, let's say, and it has these where it can be cut the same way. Turns out, then you can put a new gene in there on those sticky ends. That's what molecular biology says.
So we're replacing it.
I mean, you can insert a whole other thing in there. Yeah.
Where does the other one go?
Doesn't go anywhere.
Okay.
So yeah, so like imagine that these, I'll show you pictures better than I can draw. So we were talking about these. So yeah, so if the purple is your plasmid and you had digested your purple with the enzyme, it would leave the sticky ends of purple and purple out on the sides, right? So now you can introduce, insulin gene that has the same sticky ends on it, and it'll just stick in there in the middle of what was already there. And now you can put that plasmid in a bacteria, and that bacteria is going to crank out that protein like nobody's business. And it does, and that's where we get insulin from. So this is bacteria. So bacteria will take up the plasmids and then they'll crank out insulin. And then in their like media, their broth, we can actually purify it out of their broth. Yeah. And then you have it for shots. Now we'll get to that part in a minute. Yes.
So we use bacteria because it reproduces so prolifically?
Yes.
Okay.
We can use yeast as well. That's a possibility. It just depends on like what protein and how easy it is to deal with whatever thing with your genes or whatever, right? Whatever's available. It involves you having to know the sequence of what you're dealing with, through and through. You would have to know the sequence of your gene through and through, so you don't accidentally cut it in the middle of it. You have to know the sequence of the plasmid that you're putting it into through and through, so you don't cut it in the middle of it, and all of that stuff. So
that's why it's exciting when they figure out this gene is related to this cancer or this whatever.
It gives you a stepping stone.
Okay.
So this is just one of the things we can do with that information, of course, just the beginning. This is like where scientists would step off if they're investigating that, whereas medicine will take it kind of a different direction. So imagine that I just take somebody's genome, their chromosome, whatever, all the chromosomes in your cells. You come up here, let's just say, we're just going to put up a scenario. Somebody came up here and killed Professor Yost, okay? But Professor Yost fought back, okay? And there is a drop of blood up here, a tiny, tiny drop of blood that we almost missed that belongs to somebody other than Professor Yost whenever we did the DNA testing. Restriction fragment length polymorphisms, this is the one that I had to always think myself through this term. But imagine I cut my DNA with one of those rando enzymes, the palindrome cutting ones. It will cut at my palindromes, but where my palindromes are may not be the same as where yours are because you have different genetic code than I do. In fact, it's so reliable that you'll almost never see it even remotely close to another person unless you're an identical twin. So that's crazy to think about, right? So the patterns are always different. So anyways, restriction fragment length polymorphisms are just telling you, like, if you have this whole sequence and you have that whole sequence and they're not the same, then you can get different sizes when you cut them with that one enzyme. Okay, that's gonna come up later. Ligase, of course, if you're sticking a thing inside of, remember, don't let me say that. I was about to say stick a thing. Anyways, if you're sticking a gene inside of a plasmid, it's not good enough just to stick it there. You have to seal it up, right? And so that's where DNA ligase comes in, just like we needed with our Okazaki fragments, right? So we need to seal those up. We need to seal up these guys too. So that's why I said this is where it's going to become even more like relevant to us. We also can use reverse transcriptase, which we learned about in the virology chapter, right? Reverse transcriptase. Remember, we look at transcription, reverse of that, going from RNA to DNA. Reverse transcriptase. And that is useful, and I'll tell you about why in the next slide. We mentioned in the last chapter, this is why I said that the last chapter was so important to this chapter. We mentioned in the last chapter that genetic info of eukaryotic cells, remember only eukaryotes, contain introns and exons. And your genetic information is either intron or exon at any given point. And when you make your messenger RNA, we have to splice out or cut out the crap, the introns. You know, my stories about, you know, my conspiracy theories. And like, you know, Dr. Shearer's stories about Morse code and all those. Cut that out and put together the information that matters. That's the exons. Bacteria don't have this problem, but that also means if you just stick a human gene in a bacteria, they're not going to know what to do with that because they can't splice it. They can't put those together. This is where it gets nice because with the discovery of this horrible disease, this horrible virus, HIV, we found reverse transcriptase, which has let us take those cells from your working pancreas and get the mRNA out of it. The actual mRNA that is already cut up, that's already been spliced, that is ready to be read by ribosomes. We can get that out of those cells and reverse transcriptase give us DNA. And now we've got DNA that's already got the exons smashed together, the introns removed. Bacteria don't have to ever worry about it. It's just a working copy. And then we just, you know, you do it the one way with reverse transcriptase, you get a single strand, and then DNA polymerase can make it double-stranded. And this is what we would call complementary DNA because it's complementary to that actual functional mRNA. It's pretty neat, right? Okay. Thank goodness that we got something good out of such a horrible thing. So this is CRISPR. I don't have a lot to say about CRISPR, I'm not gonna lie, because it's so like understudied and we haven't really like been able to apply it to anything useful other than like some rats and stuff. It's just not like working out the way that we were hoping it would, but it is going to be useful. I can almost assure you of that. Just time. So these are clustered, regularly interspaced, short palindromic repeats. So usually what's going to be going on is this enzyme Cas9, which is associated with these, will go and cut out these points. But we can actually train it, it turns out, not like the restriction endonucleases, which tend to be just how they are. We can train this one, like rewrite it, to do cutting wherever we want it to. So that's pretty interesting stuff, right? It should be. If you don't think it is, you should. Of course, then this leads, well, we'll get to that later. I can't get carried away because that will really take a long time on this chapter if I do. So, all right, anyways. So originally, same thing as the restriction enzymes. This existed in bacteria to get rid of foreign DNA, and we're hijacking it in science. It will allow us to cut anywhere and genes, which is useful because we won't rely on there being a palindromic sequence and having to design this and design that. We can just cut and paste wherever we want. That's the beauty of CRISPR, except that it doesn't actually work that way. All right, because how do you do that in one cell and then have that be spread between other cells? That's the problem with it. All right, so PCR. This is one that I have to absolutely make sure that you guys are getting a good primer on, because PCR, one of the most important diagnostic tools in your lifetime, if not the most, okay? This is relied upon probably now more than any x-rays or anything else that you would see in a hospital. Because we don't just use PCR to diagnose COVID. We can use PCR to look at mutations on people. We can use PCR in the scientists in the background to discover things about the disease that you're working with in the hospital. In fact, I don't know if you can point me out a scientist that doesn't use PCR. PCR stands for polymerase chain reaction. Polymerase is referring to our good old friend that we started out with, DNA polymerase. So we're talking about making DNA in a chain reaction. This is exactly what's happening. I don't know what else to say about it. It's hard for me to word this in a way that would be more obvious, but like it's just PCR is just making copies of DNA. And I'm going to draw you a nice little picture on the board here in a second. But yeah, we can replicate our DNA replication, same thing. And we can go from, take a biopsy from somebody that we suspect may have cancer. Out of the thousands of cells that you got in that biopsy, if not millions, if there's even one in there that is cancerous, PCR can detect it. PCR is so good at its job, it can diagnose an infection from a single gene copy. That means all you needed on your little swab that they shoved way up into the back of your skull was one virus to get positive on it. That means that if you were negative, you really were negative, because it would have been able to perform its job from just one copy. The issue, of course, when we were doing PCR with COVID and having so many people getting tested is cross-contamination of, so getting false positives. Because these people that are handling all of these samples, you know, can only like be changing their gloves every so often. And if somebody else didn't handle things properly before, now you've got positive on you, and now this person's testing positive when they were negative. I mean, is that really so bad? I'm not sure that it is, but I'm just saying that's where the unreliability may come from.
So this makes it where we can take like a smaller amount of tissue or whatever for a biopsy. Like we
Yep.
have to,
Yeah, and this is very beneficial for that sort of stuff. It also means that if you are somebody who's coming in for an organ transplant and you want to get tested to see if you qualify to get put on, you know, the UNOS list, and let's say you even get, you know, approved, and now they want to start doing genetic testing on you and everything like that, they'll just take your blood, like a couple vials of blood. And that's all they'll need for all of the testing for like almost probably the next year or two. And then they'll get new ones just to be sure you're not making new antibodies, which isn't related to your DNA. Stuff like that. So we can do sequencing on your cells that we get from your blood and determine your genetic makeup to better match you with people who have a similar genetic makeup on the immunological level, so you won't be as likely to reject their organs. There's only so much we can learn about things that would make you likely to reject an organ, unfortunately. We can do all the testing in the world. I had a friend of mine, I don't know if I've ever mentioned this before. My job before the food testing lab that I keep talking about, before that, I worked at Integris as a, I don't know how to word the word. I worked in their transplant immunology lab. So as a transplant immunologist, that's a good word for it. Anyways, I was like histocompatibility technician or something like that. But anyways, I was one of the people, like there's only 6 people in Integris's lab that does this for all of Integris. And I don't know if you guys know this, Integris is the only hospital in the entire state of Oklahoma that offers like transplants outside of livers. OU Children's just started their program for heart transplants for children. Just started it. So if you need an organ transplant in Oklahoma, and it is usually state associated, then you have to go to Integris to get it because there's nobody else that does testing for it. So you can mail your samples in and we can do the testing for you. But when you need the transplant, when you get a call that you have a match, you either have to drive or they have to have somebody come and get you in a helicopter if you can't drive to take you to Integris so you can go get your heart or your kidney or whatever. And I had a friend of mine who her sample came through my lab and she'd been waiting for a kidney for a long time. And everything was clear. And I did all the testing for it. I just happened to be the person that was there testing it on it. And I was kind of excited. Of course, I didn't say anything because I wasn't supposed to, you know, and I don't mention any names or whatever. And it was all clear. And then like a couple years later, she rejected, even though everything was clear. Everything pointed to go. And it's just stuff that we just don't know about yet. So it's unfortunate. But hopefully studies that we can do, making copies of these things and why this is useful, not just for saying, hey, ta-da, this has cancer. Hey, ta-da, you have, you know, this disease. We can take a small sample of somebody and make a copy of their genes and then use those genes as like our template for studying this aspect of the human body or that aspect of the human body or whatever, without having to take tissue and tissue and tissue and tissue and extracting and extracting. We can just make copy and copy and copy and copy and copy and copy with this. We can go from one molecule of DNA, one string of DNA to billions in just like 3 or 4 hours. Incredible, incredible. You don't need even that many, you know? So it's really one of the best inventions that we've ever had. Now, PCR doesn't itself tell you any answers. All PCR does is amplify the DNA. So there's a way you can use it diagnostically to tell you if you have a disease. So you don't just like walk up to a PCR machine, pop DNA in there and press play. That's not how it goes. Because if you did, what would that really tell you if you press play? It's just going to say DNA is in here. I don't know what, because you didn't tell me to look for anything specific in a specific way, but there's DNA in here. That's pretty much what it would do if you just left it like that. But so it gets complicated. I just want to go through the basics first, though. We have to use primers. So we've already learned about DNA replication and the steps that are involved in DNA replication. We separate the strands, right? You add a little primer because DNA polymerase doesn't add on to nothing. So you always have to start with something that's already there. It's RNA usually, a couple nucleotides. And you build off of that. You go back and change it to DNA later. In the Okazaki fragments, we have to do that over and over again. So there's a lot of primers and replication just on its own. In PCR, it's that. We're taking primers and we're taking DNA and nucleotides and DNA polymerase. So we're taking all of those same things for replication and putting them in a little test tube. And then we're forcing it to do our bidding. Because those primers that we have, we can actually by nucleotide, design them. If you know the genetic code for the spike protein on your COVID virus, right? You can design primers that will detect that gene that are so specific enough, they won't bind with anything else that might be randomly in somebody's DNA or in a virus or anything. So that when you put those primers and those only bind on there when it's there, and then you build off only when those primers are bound, then you're only going to build your DNA when you have COVID there. Yeah. Makes sense? So that's how that works. You have to have the primers first. So if you want to test for Ebola, got to get the primers first. You can't just snap your fingers and it tells you. And you can't do multiple things all at once because the way the machines work, they don't know what's going on, the difference between them. So you have to do them one by one. They have designed some systems that can do like up to 3 now. You have a PCR machine that can detect in one. I don't think it is one. I really don't think it is in one vial. I think it's in separate vials still. Somebody tried to convince me that I still think they're in separate vials. Which is what I did at my work. I could put like one tray in there and each one was something else going on. But anyways, you can test for influenza and COVID and RSV from like one sample. So it's great. It's great that you can do that, but you have to have the primers.
Where is the primers come from?
You make them. So, so you make them or you order them, right? So nowadays you can, if you, like I said, if you knew the sequence of COVID, let's say, and you're like actually somebody who's studying it, you know the actual ATCG code, you can pick an area that you think is good because you would understand, you know, what's better for the sequence. Then you call up a company and you say, I'm emailing you this primer code and I need, you know, this much of it. and they will literally make it for you with their machines, their kind of old machines or whatever. This is how much I know about that part of
It's
it.
all just AGTC
Huh?
or you, okay.
Huh? Yep, and then those primers, they'll send them to you, and then you can put those individually into your mix. Now look for it. But if you don't know the sequence of the thing, you can't PCR it. So if somebody hasn't studied it well enough, not everything has its genome mapped, you know? So some things don't have genome mapped, and we can't use primers to look at it. And if you have somebody that's actively dying of Ebola, for example, you're not going to wait on the CDC to send you some primers or for you to send the sample to the CDC and then test it. They'll test it, but you need to do something before then, right? So it's that sort of stuff. Like, it's definitely useful, but it's not 100% reliable. There's just stuff. So we already know we need the primers and the polymerase. The problem is what we're going to do to separate, just like our very first slide in this, we're going to heat up our DNA. We're going to heat it so hot. It's about, I think, 94 degrees Celsius. It's almost boiling. So hot to separate all of that DNA. And then, unfortunately, we call that denaturation. Unfortunately, though, proteins also get denatured if you're heating them up that high. And what is DNA polymerase? The protein. So what we do is we found some bacteria that are in like a deep sea vent that is like, you know, super, like the hydrothermal ones, right? The ones that are like super hot. And their DNA polymerase naturally is heat resistant. So, I don't know. There you go, we stole it. We made it useful. Anyways, we have the steps for PCR here. It's only got 3 steps. So it's really not that bad. You denature, which just means heating it up and separating the strands, okay? So imagine it like this. You've heated it up and you separated your strands, right? Now you can add on your primers. So you're going to cool it down a little bit because you can't just add on primers in that hot because it separated these. Of course, it's going to have separated primers. So we're going to cool it down so that your primers can come bind on, that you so carefully designed. And then your DNA polymerase can do its work, adding on nucleotides from those primers. You know, complementary sequence or whatever. So denaturation is your first step. That's just separating the strands with heat. Priming is cooling it down enough to let your, or I guess they call it annealing, priming, it's the same, whatever. But anneal just means to get them to stick together. So priming, we're going to cool it down to anneal those primers onto the appropriate sequence. Then the last step is extension, which is just adding on the nucleotides. That is literally all PCR is. So not too bad. This is what you would end up with, right? Basically semi-conservative replication again. Just even like, I feel like even easier. So this is 1 round of PCR. So each round of PCR doubles what was the previous round. So 1 round, you'll have 2. You went from 1 to 2. After 2 rounds, you went from 2 to 4. After 3 rounds, you went from 4 to 8. After 4 rounds, you went from 8 to 16, and so on and so forth, whatever. It's going to get weird and the numbers are going to get confused in my head if I keep doing it. So you will have a test on the lab exam about this because this is also clearly like lab related stuff. So it will be asking after this many cycles of PCR, how many would you have if you started with 1? It's not going to be 97, okay? It's going to be like 4 or 5, maybe 6 tops. If you can double your numbers, you should be fine, okay? So yeah, just warning you so you know, but it's really not that complicated, right? We're just making copies over and over again. That's all it is. Okay, so I never do good with the timing of this lecture. Right, so when we're cloning something, we're basically taking the genes from a donor, DNA from a donor, and inserting it into a vector, that's like our plasmid or whatever, and then taking that plasmid that we manipulated, that vector, with our gene, and inserting it into a cloning host. So taking human insulin gene, so that's our genetic donor, sticking it into a plasmid, so now it's plasmid plus insulin gene, putting that into a cloning host, so let's say bacteria, E. coli, and now it can just crank out insulin for us, okay? So we already talked about some of the ways that they do this, like making the complementary DNA and using PCR and stuff. So some of this stuff we'll kick through a little bit quicker, but plasmids, I do want to remind everybody, these are DNA, small circles of DNA. They have extra genes that can help the bacteria. It's beneficial for them. So plasmids, as well as bacteriophages, we can hijack that system and make it work for us. Because you guys remember how bacteriophages will insert their little genes into the bacterial genome for, like, what was it called again? Lysogenic conversion. So when we have that happen, we can also hijack that mechanism and make them insert genes that we want them to. So we can do that. So here's an example of how we might look at one of those plasmids that we would work with. And one of the things that I want to point out here is the brown one up there, AMP. So that AMP-R is ampicillin resistance. So if we can put this plasmid into our E. coli, we're not just picking up E. coli off the ground, right? We're buying it from Bio-Rad or something. And that pure E. coli strain, like the ones we work with in lab, doesn't have ampicillin resistance naturally. You have to give it that. So this plasmid has that in it. Also, you can see up there, we have all these EcoR1, SAC1, KPN1, all those are restriction endonuclease sites. So if you know you can work with this code that you put in on your gene, and you know it has a matchup with this plasmid somewhere, you know you can use that enzyme on both of them, for example. Then you can insert your gene in there and it will interrupt that gene, the blue arrow gene, whatever that is. I don't know what it does, but it doesn't matter. But it'll interrupt that. So you will know not only that that gene has been interrupted because it won't be doing its whatever anymore, but if you plate your crap that you put with your plasmid and your bacteria, and you made your bacteria take it up because it's outside of the cell, and that is a process that we already learned about that is called transformation, force that to happen, then plate it on a plate that has ampicillin on it. And only the ones that took up that plasmid should grow. And that is genetic engineering. I don't know what else to say. So there you go. We'll learn more about this in detail in the lab. I think it's like one of the last labs, like 10.2 or something like that. You can use this technology, and we do use this technology to make a lot of treatments in human medicine right now. You can even mimic other proteins, not just insulin, but even immunologically related proteins that can help treat other diseases, and there's all sorts of stuff. But insulin was one of the first ones. That's why I like to go over it. And we already talked about this. Now you're going to insert it into your little plasmid. You're going to cut it, and then you're going to put your little gene that you made and put it inside of your plasmid. And then you're going to put your plasmid inside of your DNA via transformation. Then grow it. It's going to make the product, and you can purify it out. Okay. There's also something called synthetic biology. Synthetic biology is when you play God, literally. So taking just nucleotides and generating your own sequence based on what you know about genes and what genes would code for blah and blah, 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 what you would need for a living organism, and then designed your own based on that. Not from using cells or anything, literally only from nucleotides. People have managed in 2010 to just use A, T, C, and G to make a brand new organism. Bacteria was still young.
Is that good?
I don't think so. It doesn't feel right. It feels questionable at best, right? So yeah, I mean, I agree with you. I'm with you on this. So we can also use this process, the PCR process, to look at maps of genes and genomes and all of this. We can even take random primers from a species that we've never studied before. We can put those random primers with their genes and just hope we get a sequence out. Hey, you get a sequence out, now you can design primers based on literally that sequence and just start shotgunning their whole genome. And you can figure out a whole genome that way when you do nothing about that organism to begin with. It's pretty crazy how you can use PCR and sequencing, which is a different thing. But that has to do with adding a nucleotide and running it on these gels. Because they used to do it in actual gels, and it would be like not like the CSI gels. It was like massive gels that would have literally just tiny minuscule amounts of stuff based on like how long your thing was from like one nucleic acid to another nucleic acid. God, I don't know how they did it, but I used to have to pay a lot of money for them to do it, and they would always complain that my samples weren't pure enough. So whatever. So you have your DNA and you need to separate it out. Let's say you were wanting to get your insulin gene from somebody, or you're wanting to see who killed Professor Yo's stuff here. So you might even subject that sample to those restriction enzymes, as well as everybody in here, your samples to the restriction enzymes. But now what? Cool. We've broken it into parts. So that's what gel electrophoresis is for. You get a pattern of DNA fragments. It's a gel. It's called agarose gel. And it does come from agar. It's a sugar that's based from agar. It's just purer, but it has a very similar texture. It's almost clear. Anyways, it has these wells at the top. And I would digest my sample, or not me, I'd be dead, but they digest this little dot up here, and we digest all of you guys' samples that we took from the swabs in your cheeks and PCR'd up just to be sure we had enough of it that it made any sense at all. And then popped it in to these little wells on this gel and turned on some electricity. There's liquid in there, of course, too. It's helping everything move, but and keep it all happy. But DNA, as we've already discussed, has that backbone, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, over and over and over again. All DNA is like that. So what charge does phosphate have? Negative, right? Very negative. So that will move to the positive end in your electrically charged box. So DNA will migrate based on charge through this gel. But how does that actually separate out those bars of different sizes? Well, just like you could imagine, if you're tiny, like I said on the thing earlier today, which I know a lot of people on it, and that's fine. My example was, if you need to climb through a pile of rocks to get to the other side, there's rubble, somebody like bombed this place, and we need to get on the other side to get to our kids or something, right? You got to crawl through the rocks. Who's going to make it faster? The cockroach or the elephant? Cockroach. That's this. So the tinier bits of DNA are going to move faster, and the bigger bits are going to get caught up. So now we can separate out the different sizes. Make sense? Based on the sizes that you get when you cut with those enzymes. Look at that. So if we were looking at this exact example, we could say there are some in here that look kind of similar, sure. But we have to point out number 3 and number 4. Tell me that's not the same person. It's got to be, right? Look at the difference with the other ones. So anyways, it's basically as simple as that. It's so different, like I said, you can pretty much rely on that to work for you. If you knew a sequence that you were looking for, let's say you have a tissue specimen and maybe even a histological specimen that you're going to keep intact, or you have thin slice, or you're dealing with bacteria, you want to know if they have a certain gene in them. All of this, if you know the sequence of that gene you're looking for, you can create a probe with it, and it could be fluorescently marked, or it could be other marked. I don't know what, anything else. But a lot of times fluorescence, right? So it glows. So if it sticks, you'll have your sample glowing. So we can literally take a tissue sample and put that onto the tissue sample and see which cells in that tissue sample match up with the DNA that you're looking for. So if it is cancer DNA as opposed to the normal DNA, we can see it glow on that tissue sample. That's DNA hybridization. There's a lot of different versions of it, but that's just one of them. That's called FISH, which is fluorescent in situ hybridization. This is all this is. If you're interested, you can go through the details. Here's a bacterial version of it that goes step by step through it. I would have normally go through this, but I always talk too long on this talk. So guess what you get? You get me skipping through the questions. All right. Because this chapter is so interesting and people always have so many questions and want to discuss, and I just always have taken so much time with it. Even though it's like one of the supposed to be shorter. But anyways, when we talk about the size of DNA, we're talking about those fragments are separating out based on their size, right? How many bases are in them? So whether A, T, C, G, those are bases, each one is. And when we talk about base pairs, if this was on this green, A, T, C, G, And this was whatever the opposite is. I don't remember, wasn't thinking through it, but anyways, the opposite on the other side, it's still a base pair. So we say that's one base pair, two base pair, three. So you can still think of it as bases. Okay, base pair, base. Don't get confused. They're not. Don't worry about it. No need to separate, honestly. Not in this class. So we can also, because you're obviously your genes, and I'm telling you, your genes, you have 20,000 genes in your body, and each of those genes is going to be thousands of nucleotides long. We're not going to count it in single nucleotides. So we can count by the average gene in E. coli is 1,300 base pairs, or we say 1.3 kilobases. So kilo is still talking about 1,000. And mega is talking about 1 million. I just realized something. It's not important to you guys, but it is to me. So I just realized it, and that made me happy. Okay. Anyways, you do need to know this, because if I ask you guys on a test, which one is the smallest, and it says 1 kilobase and 20 megabases and 400,000, or I don't know, whatever base pairs and whatever, you need to be able to do the calculations between. And it's usually not even that complicated, and people still often get it wrong because they'll just see the smallest number and pick it. So just be careful, okay? Our DNA, we already talked about DNA profiling. That's really what we were talking about with our cutting at the sites and making the different bands and then looking at who was a murderer. So we can look at that way and find out which matches up with our suspect. So, and I already went over sequence maps, doing all of these things to figure out the genomes of things just randomly. This is what I was talking about with those random sequences. You can build it up, even a couple nucleotides down and line it up, and then suddenly you have one code, and it's all long, even though you got it in pieces. That's how genome sequencing works. It's high throughput genome. And that's all genomics and bioinformatics. Okay. We want to understand our genome so that we know where our genes are, but we also want to understand the different versions of our genes, right? Not just that this is a gene that codes for hair color, but that this codes for this hair color because of this reason and whatever. And obviously more important for diseases. So there are these things called single nucleotide polymorphisms. That, don't freak out from the word, single nucleotide, just one nucleotide. Polymorphism means changes that they're going to be causing, okay? So it says the human genome contains 10 million single nucleotide polymorphisms. What that is saying is that you can have one nucleotide change in your gene, and that might make it just a different color of hair. Or that one nucleotide change in your gene could kill you, depending on the gene. So we want to map out all the different versions of each nucleotide that could change all these different things. As long as it's just one nucleotide changing and causing a different version, that's single nucleotide polymorphism. Of course, you can have more than one thing going on, but. So thrombophilia is the example that it gives us. Thrombophilia is a clotting disease. It's increased clotting. You might think like, oh, okay, so they just clot a lot, who cares? But it's actually an extremely serious thing, especially as you are aging and if you're not eating healthy or taking care of yourself or taking blood thinners. So one nucleotide change in their genetic code, just one nucleotide, and they have this disease. And it's crazy to think about. But yeah, this can be very seriously associated with having a stroke at an early age. I don't know if you guys know, but if you have not worked in a hospital ER and you have not dealt with somebody who possibly had a stroke, you have no idea how seriously they take it. It is insanely serious. And the lab will too. And I can attest to that. They will call us up and before they even get the patient. And they will say, we have a code stroke. And we have to get everything ready so we can move everything as fast as possible to treat that patient as fast as possible. Because if you can get them treated within the golden hour, they're way more likely to survive. And it's a very serious thing. So we take it very seriously. Integris takes it very seriously. I can say, I don't know about Baptist. I was working at Southwest then, but they were always very good about contacting us. And so clotting is a serious issue. It's not just you just clot some. You don't want to get strokes. That's a horrible thing. So DNA microarray analysis, of course, we're getting close to the end. I feel like we are. This is like if you take a microchip, essentially, but it's a little bigger than that, and you have 365 little dots on it. And each of those dots has a different version of cancer, let's say breast cancer. So your patient has breast cancer. what kind, right? What gene? So you can put all the different versions of breast cancer genes on that one thing, put your patient sample on it and mix it up for a little bit, pop it into the machine, and it'll see which ones are glowing like the ones before and tell you what genes are affected that way. Very high throughput way to look at a lot of different possibilities if you aren't sure how to narrow it down with your patient. Very expensive as well. That's just saying about it. We already talked about kind of the uses of this stuff. I don't need to go about it, but I will say you can even do things like creating monoclonal antibodies that will target certain things. So this is a thing that I want everybody to understand. We're going to get to it in immunology, but since we're here now. Okay, in immunology, if you are having any inflammation whatsoever, stub your toe, you know, your back just hurts that day. Any inflammation of any kind whatsoever is controlled by your immune system. And if you are having an issue with inflammation, and one of the things, I mean, it just depends on what disease we're talking about. A lot of these are going to be autoimmune related. Your body's attacking itself, so inflammation, right? It's usually the inflammation that's causing the problems. So with Remicade and Humira, what they've done is created antibodies. Let's just say, the blue thing is the antibody. The blue is the antibody, okay? Normally, your cell sends signals saying, time to cause inflammation, and the other cells in your knee or your toe or whatever are being activated by that, okay? Saying inflammation time, great. What these do is they create an antibody that binds to the receptor on those cells that normally this would interact with, not can't. That seems like a great thing. Oh, no more pain and no more inflammation, except that those things exist for a reason. And that's why if you're on Remicade or Humira, you have a significantly decreased immune response. Because you're supposed to have that, okay? The things the more you know, that's what I say. You guys don't know what I'm talking about with that. Anyways, there's a lot of stuff you can do with this. This is just talking about other possibilities that you could learn about with it. GMOs, these are some of the things I do want to talk about. GMOs and definitely gene therapy. So GMOs are genetically modified organisms, also known as transgenic organisms. And okay. Anytime that you have any change from one organism's DNA with another's, that is a GMO or a transgenic organism. So those bacteria that make the insulin, obviously, those are. Guaranteed any corn that you've eaten probably in the last 10 years is guaranteed. 98% of all strains of corn grown in the United States now are, in fact, GMO. And of course, they have to label that on your, cans of corn or whatever. Most people don't check. And, I don't know what else to say about that. But so introducing these genes, what they do with the corn is they have taken genes from bacteria, Bacillus thuringiensis, and they take it's like it bites off. They say destroy invading insects by modifying Pseudomonas florensis with that. But now they can take that and put it into corn. So they call it BT corn. And they've realized that they can fight off insects and not have to treat the corn with pesticides. And isn't that a great thing? Except that the corn that isn't getting pests on it can send its genes to other plants. And then those plants also will be resistant to pests. And suddenly you've got an ecological problem on your hand. And there has been evidence of that happening occasionally. This doesn't happen in animals, by the way, just before you start freaking out, right? Not even animal to animal, which is why I bring this up, and I need you to be sure that you understand, okay? So here's the issue. Yeah, it's usually going to be an ecological problem. That's the real problem with GMOs. And the unknowns of it, of course. If you're changing something's genetic information to say this thing, how is it going to impact that organism? And how is that going to impact that organism, how it relates to us or the ecosystem or anything? Can you really know that? You know, of course, they're not doing those studies. Or would they? So here's the thing that I want you to know if you read this class and learn anything about a GMO. I don't care who you talk to or what you've read or where you've gone or whatever. If you go to actual reputable scientific sites, if you are somebody who is of the mind that you can eat a piece of chicken that is GMO and it could potentially cause your body to mutate, you are absolutely wrong. I'm out. Okay? Here's what I'll tell why. And I'm not attacking anybody, I just want you to be educated about it. be upset about GMOs. I am too. I don't think that they were responsibly used, right? But that's not the reason to be upset. If you eat normal chicken, does your body normally incorporate that chicken's DNA into it? No. So why would it do it with the gene? It's not special DNA, it's still just DNA. So it's not going to do it with that either. What happens when you eat a chicken is, any chicken, GMO or otherwise, your body's going to digest down that DNA and use the parts of the DNA to build up other things in the body. It don't matter if it's GMO or otherwise. DNA is just DNA. It's not different because it came from another organism. So anyways, that's your lecture for that part. That's my conspiracy theory of the day. It's not really right. So we're going to talk about gene therapy, and I want to show the picture because it's the better way to explain it. Gene therapy will take out the working gene from somebody that has a healthy gene, and by the ways we already talked about, put it into the genome of a virus. And we can use exactly the genes that we want in that virus and take out the ones that we don't want to keep. And cause that virus to insert our gene into your DNA. So taking HIV, for example, clearing it out of this HIV stuff, but still making it insert the genes into the DNA of the person, right? This is real life. So you can do this. Take out somebody's bone marrow, mix it up with these derived viruses, and put it back into the person for an infusion. And that can spread that virus actually throughout the body and replace that gene. So they have a functional version of that gene. So now they don't have to die. Legit. This is permanent. So we have, as of 2022, which is when this book was written, we have approved therapy for 13 different diseases, including spinal muscular atrophy, inherited vision loss, and acute lymphoblastic leukemia is what that's supposed to say. Imagine that. So this is somatic cell gene therapy. It's not heritable. It's not going to change like your gametes or whatever. Those are already like doing what they're going to do. There's not, unfortunately, anything we can do for that right now. But it will change all of you. And if you did have kids and they did happen to get that, now you know that they have an option for treatment. There is stuff in the works, though, to work on germline therapy, to insert genes into the eggs, the sperm, embryos, and stuff like that, so that can be heritable. But don't forget, conspiracy theory number 2, don't forget that this, if you have not seen Gattaca, y'all, this is the slippery slope, right? Cure the diseases, okay? So we cure the deadly diseases that people have, the ones that are just, cause inability to walk or, whatever it is, disfigurements and disabilities and everything like that. So what else should we cure with this? Oh, well, this person, they had the lazy eyes. Now they don't have to have that problem anymore because of this. So, okay, well, now I wanted my baby to have blue eyes, though. Where do you draw the line? You see, he's like, okay, that sounds kind of cool. You have designer babies. Yeah, until I can afford to have my baby be smarter than yours. Now what? Out there is that. When mine gets to go to the Ivy League school just because I chose the genes, because I could afford it. That's eugenics, if you haven't heard of that. This is the slippery slope. Is it good? Sure. But like where do we draw the line? If you haven't seen Gattaca, I have to keep bringing it up. If you haven't seen it, look at it. Because it's an old movie, but it still checks out, okay? You can use little bits of RNA to silence DNA sequences. If they match up to the DNA sequences, they can just bind to it and the DNA can't do its thing. I promise we're gonna go. I really do promise. This is it. Yeah, that's it, right? That's the last one. Okay, cool. Okay, oh, thank God. Okay. Lab. I hope that was at least interesting for you guys, even though it went over. The lab will not go over. The lab is pretty quick.